New admission. School. Where is it? Okay. You from that far. Okay. Now, where was it? Which topic we were doing? Heredity. I think I gave you one question on the application of the law of segregation. Did I? Check. Yes. Check karo. Last class we had discussed on the law of segregation, law of dominance and law of segregation. Yes or no? Yes. So uske baad we had understood the phenotypic and the genotypic ratio, and after that I gave you one question. Did I give you a question? Check karo. Yes. Kiya homework? Yes. You? Busy? शॉपिंग वॉशिंग क्लीनिंग सब तुमको ही करना पड़ता है। कितने लोगों ने होमवर्क किया? कितने लोगों ने होमवर्क किया? हाँ किया होमवर्क? क्यों? 10 मिनट नहीं निकाल पाए इसके लिए ओनली 10 मिनट्स का काम, 10 मिनट्स भी नहीं 5 मिनट्स का काम है। 20 मार्क का एग्जाम के लिए कितना क्या तुम पढ़ रहे हो? 40 मार्क का एग्जाम है। ये एक्सक्यूज थोड़ा पुराना हो गया अभी, ये सब एक्सक्यूज अभी पुराना हो गया। कम विद समथिंग न्यू। फटाफट करो, जस्ट 2 मिनट्स में फटाफट कंप्लीट करो। करो। What are the steps? Law of segregation. The question that I gave you is based on the concept of law of segregation. What was the question I gave you? Read out. What will be the phenotypic and genotypic ratios when? Of the F2 generation. Of the F2 generation when? A homozygous plant or round seeds are crossed with plants with wrinkled seeds. A homozygous plant with round seeds. So what are the parental combinations we take? The parents, what will we take? Round and wrinkle. Round, the parent will be homozygous round or heterozygous? So this is an application of the law of segregation. The law of segregation. And what is it? I gave you a question that the parent plants. What about the parent plants? Phenotype क्या है? Phenotype क्या है? Round seeds. How will you write down the genotype? Capital R, capital R. And cross with what type of plants? Wrinkled seeds. Wrinkled seeds. Cross with wrinkled seeds. What will be the phenotype? Smaller, smaller. Smaller, smaller. Now keep going. What are the next steps? I'll have to find out the gametes. Next is you will have to find out the F1 generation is what? F1 generation plants what? After F1 generation plants, you have to again find out the gametes of F1. Then the F2 generation, and to find out the F2 generation, you will go for the crisscross method or the Punnett square method. Karo. See the previous cross and keep doing. See the previous example, it is exactly the same. See the previous example and keep doing. आया समझ में? How many of you did not understand absolutely? How many of you did not understand this concept? What will you write now here? Gametes, what will you write? Gametes you will write. One type of gamete hai to one time also you can write, or we write twice. What do we write here? Gametes, always encircle. Next. Next. F1 generation. What to write? Fertilization between this and this. What will be the pair of chromosome? Now that I write capital R and small r, this is known as genotype or phenotype. Genotype. This is known as genotype or phenotype. Genotype. What is genotype? The pair of allele that you write. The combination of allele that you write in letters, usko bolenge genotype. What will it give rise to? Now, out of these two, which one is dominant? So, what will be the shape of the seed? Round. So, we will write this is hybrid round seed. This is known as phenotype. This is the phenotype. How does the seed, plant, flower look like? Phenotype. What is the next step that we have to do? Next step क्या है? नहीं, gametes के पहले एक और step. I want to get the F2 generation. What am I supposed to do? Cross with another F1 plant. Cross with another F1 plant. So I take another F1 plant, and it is understood that the plants of this generation will have the same genotype, same phenotype. So I write hybrid round for this also. Hybrid round seed. Now, how many gametes from this plant? How many types? Four. How many types of gametes from this plant? Two. Sure? Yes. How many types of gametes from this plant? Two types. It's a heterozygous parent. Two types. What do I write here? Same, because the F1 plants are understanding? Yes, ma'am. F1 plants are heterozygous. Ab next kya karna hai? If I go by the crisscross method, what am I supposed to do? I will just number them. And then I go for the process of fertilization between 1 and 2 or 1 and 3. 1 and 3 and 1 and 4. So, fertilization between these two gametes will give us what? A pair of chromosomes like this. What do I write? Pure tall, pure round. Now, this is pure round seeds. I take 1 and 4 now. Hybrid round. So, I write here capital R, small r, and this will be hybrid round. This will be hybrid round. I take now 1 is over. Now I take 2. 2 and 3. What to write here? Capital R, small r, and we will write hybrid round. What next? Small r, 2 and 4. Small r and small r. So we will write pure wrinkled seeds. Pure wrinkled seeds. Clear? Yes. Now, how many types of phenotype in these 4 plants? How many types of phenotype? Two types. Which are those 2 types of phenotype? Round and wrinkled. Round ya to wrinkled. 2 types of phenotype. So, how do we write down the phenotypic ratio then? 3:1. What is the phenotypic ratio? The phenotypic ratio would be 3:1. I write 3:1. Why 3? Because out of 4 plants, 3 are with round seed, then only 1 wrinkle. How many types of genotype? 3. 1, 2, 3. 3 types of genotype. So what do we write as the genotypic ratio? This is known as Genotypic ratio, what will we write?
1:2:1.
1:2:1. That is the genotypic ratio. We studied a concept known as monohybrid cross. What was monohybrid cross? The other day I explained you law of segregation, and I told you that the type of example, this type of crosses, they are also known as monohybrid. They are also known as monohybrid. What is the meaning of monohybrid cross? Why are they known as monohybrid? Why monohybrid? Because we are studying the inheritance of only one character in generations. How many generations we are studying? 1, 2, 3. In 3 generations, we are studying only one character. Which character? Round character hai. Which character? What is the character we are studying?
Shape of the seed.
Shape of the seed. And grades क्या है?
Round
and wrinkled. Name the character. Shape of the seed. 7 characters we discussed, no?
Height of the plant. Shape of the seed.
याद कौन रखेगा?
Color of the plant. Color of the seed.
Position of flower.
Position of
Shape
flower.
of the seed.
हाँ, तो ये 7 characters याद रखना है. Clear? Is this done? Monohybrid cross. For monohybrid cross, we had learned 2 laws. One was the law of dominance. What was the statement of the law of dominance? What was the statement of the law of dominance?
Contrasting alleles in the F1 hybrid plant, the alleles that
Suppress the recessive.
क्या हो रहा है? One.
Dominant.
Out of a pair of contrasting alleles in the F1 hybrid plant, the allele that expresses is dominant and the allele that remains suppressed is recessive.
अभी अपने language में explain करो.
So basically,
You are talking about this generation plants. In this generation plants, how is this? How does the law fit in? In this generation plants, क्या हो रहा है? There are 2 contrasting alleles for a gene. Only one allele is expressing. The allele that expresses is dominant. The allele that remains suppressed is recessive. Okay? Even for this concept, the second law, even for the second law also, we are focusing on the F1 plant itself. And what was the statement? The 2 contrasting alleles in the F1 hybrid remain in 2 distinct chromosomes. They do not blend to form anything new, and they segregate during gamete formation. An extended explanation is that is the reason we are getting this type of plants. Otherwise, we would not have got that 25% plants which are homozygous recessive, whichever character you speak about. Exam में इस तरह के question straight away question मिलेगा where you are supposed to write down for 5 marks. State the law, explain with an example, and certain other 1 mark question. Sometimes Punnett square asked, sometimes not asked. And sometimes you will also get assertion questions on this, some other questions on this. If this is clear, then understanding assortment will be better. Understanding assortment will be easier. Any doubts that you want to clarify first on this, tell me. Hai koi doubt? No? So then we move on to the third law. And what is the third law? The third law is law of independent assortment. What is this known as? The law of independent assortment. What is the meaning of assortment? Assortment matlab segregation, arrangement, I would say. Arrangement. Now in a proper way, arrangement. Now we are going to understand arrangement of the gametes. The concept of arrangement of the gametes is what we are going to learn in this law. Now here, let us start with the basic Mendelian concept. So parent plants. What are the parent plants that Mendel selected? The parent plant that Mendel selected was round and wrinkled. Round and no, that was yellow and round. We start with this. round and yellow seeds. Round and yellow seeds. So ये जो round and yellow seeds I am writing, this is the phenotype or genotype? Phenotype. So genotype, how do we generally choose? The letter, capital, based on the dominant always. So I would write capital R, capital R, capital Y and capital Y. Clear hai? It is like that these plants have got cells, and how are the cells? The cells would be something like this. Cells में paired chromosomes hai. In one chromosome, the condition is something this way. Capital R, capital R, capital Y and capital Y. आया समझ में? So that's why we are writing RR and YY. Is it a homozygous or a heterozygous condition? Homozygous. Dominant or recessive? Dominant. So this is understood. Now we have to write down the other contrasting plant. We'll write down the other contrasting plant. What is the other contrasting plant? Round and wrinkled, yellow and green. So we will write down now wrinkled and green. Wrinkled and green seeds. So lower case letters we will take. Small r, small r, small y, small y. Gametes, how many types? Gametes, how many types? Homozygous condition hai. It was like this just now. I showed you. Capital R hai, capital Y hai. Understand why we write this way? So it is a situation like this in a pair of chromosomes. Now it is a process of cell division. The chromosomes are going to segregate. So chromosome agar segregate hoga, then what should be the situation? Is it not something like this? So how many types hua? How many types? How many types?
Two. Types are only two.
How many types? नंबर हो गया 2. टाइप्स कितने हैं? कहाँ पे 4 टाइप्स दिख रहा है?
2 types.
How many types? टाइप समझ में आता है कि नहीं आ रहा है? टाइप्स मतलब क्या? वैरायटी. ब्लॉकेट कहाँ पे है? कहाँ पे ब्लॉकेट है? So now I'm not remembering the entire concept of genetics. ये जो हम process और ये सब examples सीखते हैं, इसमें नंबर से कोई मतलब नहीं है. Is it that when 2 plants are crossed, only 4 plant कभी होता है क्या in reality? Hundreds and hundreds of plants होता है. When 2 plants are self-pollinated, cross-pollinated at one go, 20 plants are formed. So हम why are we writing 4? We are only talking about the types here. Did you understand what I'm asking you? How many types of gametes from this pair of chromosome when they are segregated? Cell division जब होगा, the pair of chromosome will split. One chromosome, one gamete, another chromosome, another gamete. So कितने types कितने हो गए? One type. And always to remember, even if you get any other question, a homozygous parent always gives rise to how many types of gamete? Always one. Homozygous dominant है, homozygous recessive है. How many type of gametes will it give rise to? Always one. Heterozygous है तो there is a difference in the number. 2 हो सकता है, 4 हो सकता है. That's true. So I would write what type of gametes? This is 1. I can write twice also because 2 chromosome hai. If I write once, then also that is correct. Next is, what do I write here? Small r, small y, small y. Next step क्या करना है? Fertilization to give rise to the F1. So what will be the pair of chromosome form? The pair of chromosome will be this and this. This is one chromosome and this is one chromosome coming together. Phenotype क्या है? Phenotype क्या है?
Hybrid round yellow seeds.
Round yellow seeds. Understanding?
Yes.
Now next is After this, here we have to understand how many type of gametes. So पहले तो वही process है. Now that I want to get the F2 plants, what should I do? Cross to F1 plants. Cross to F1. To cross to F1, how? This is the other plant I take, and this is also hybrid round and yellow seeds. This is also hybrid round and yellow seeds. Now we try to understand how many gametes from this. To understand how many gametes from this, first of all, this is only for explanation. This does not go in the writing when you are writing this. Let me talk about this particular pair of chromosomes. What are the alleles? Capital R है, small r. Capital Y है, small y. Yes, it is. This pair of chromosomes, first of all, before it goes for the process of separation, this particular chromosome will show duplication of chromosome. Chromosome duplication होगा. So now that the process of duplication happened, now if I write that this is another duplicated chromosome, will the alleles remain exactly the same?
No.
You नहीं?
Yes.
Duplication है ना, तो alleles will be exactly the same.
Right.
One chromosome forming a copy, that is called duplication. One chromosome forming a copy, alleles भी exactly same रहता है. आया समझ में? So what alleles should I write here? I will write here capital R. What alleles should I write here? I will write here small r. What allele here? Capital Y. What here? Small y. Yes? Let me do it again. This is a pair of chromosome I told you. What are the alleles? एक में है dominant, दूसरे में है recessive. अब duplication. Now that the process of duplication happened, what are the alleles here? Capital R and small r. What are the alleles here? Capital Y and small y. But whenever we talk about the process of duplication between these two chromosomes, as a result of duplication, before the process of duplication is all over the length of the DNA, what happens? The two newly formed chromosomes, they always have a tendency of showing or crossing over. They always have a tendency of showing exchange of the chromosome parts, a process known as crossing over. तो कैसे होता है crossing over? ये हो गया parental chromosome. अब जो ये duplicated chromosome है, they will always show a process of crossing over, resulting in the formation of a X-shaped configuration like this. तो alleles क्या हो गया? Now this is capital R. Where is capital Y now?
Small r.
राजदीप इधर आ. कैपिटल आर कैपिटल वाई. अब ये हो गया कैपिटल आर. वेयर इज कैपिटल वाई? यहाँ पे. सो आई विल राइट कैपिटल वाई यहाँ पे. ये हो गया स्मॉल आर. स्मॉल वाई कहाँ? यहाँ पे. एज अ रिजल्ट ऑफ द प्रोसेस, ये प्रोसेस को क्या बोलते हैं? दिस पार्टिकुलर प्रोसेस इज नोन एज क्रॉसिंग ओवर. But during the these are not fine threads that we are talking about in the nucleus, the process of crossing over of chromosomes. As a result of crossing over, when the two chromatin threads, they are overlapping each other, they wind and unwind and get separated. So as a result of that, at this point of connection, they generally get broken down. They get broken down like this, and then इस तरह से connection or the two chromosomes this way now they get joined. पहले this was one chromosome. Now at the point where they were connected, they get broken down, and two chromosomes from two parts from two different chromosomes they fuse together. अब सेल क्यों करता है? Nobody knows. That is the behavior of the chromosomes always, and that is the reason why we are different from our parents. This is a process which is constantly happening in the chromosomes, and this is what is responsible for showing new characteristics. Though we inherit chromosomes from our parents, why are we not identical to them? because ये जो process है crossing over, as a result of crossing over, we start showing new characteristics जिसे variation बोलते हैं. अब ये तो मैं एक particular chromosome और एक particular crossing over बता रही हूँ. In the entire length of the chromosome, इस तरह के crossing over at multiple positions में होता है. And every time ये chromosome part getting connected with this getting connected with this getting connected with this is what happens. Otherwise, we all would have been exactly identical to our father, to our mother. But we have something around 10-20% characteristics जो not with our mother, neither with our father match होता है. And it is absolutely because of these type of processes. So now, when I say that what happens to this newly formed or the duplicated chromosomes, they will always undergo the process of crossover. And ये जब crossover होके when they detach, do they remain as it is like before? No, एक के साथ दूसरा दूसरे के साथ they get connected and ultimately a new combination of alleles get formed. A new combination of allele get formed. Was the chromosome like this earlier? No. Capital R and capital Y पहले एक साथ था. Now that the new chromosome thread is formed, I should write now the new chromosome or the after the process of duplication. क्या हुआ? After the process of duplication, the arrangement of the alleles have changed. What is the arrangement of the allele now? Capital R and small y. Parental में ऐसे नहीं था ना? What about the other one? Small r and capital Y. So this is what is known as exchange of chromosomal segments, जो crossing over से ही होता है. Now, if I have to tell you that how many gametes will this plant give rise to? 4. यही 4 है. 1, 2, 3, 4. So this particular pair of chromosome, which was the original chromosome we knew, 1 and 2. यही pair था ना पहले. This was the pair earlier. They got duplicated, crossover हुआ, उसके बाद split and separation हुआ. So now the pair of chromosome has become like this. So how many gametes now this plant is giving rise to? 4. कौन से 4? 1, 2, 3, 4, जिसमें 1 and 4 तो parental ही है पहले जैसे. Only 2 and 3 these are recombinants. Recombinants मतलब new combination of alleles. Parental combination है नहीं. Now they are having new combination. इसके साथ ये, इसके साथ ये. यहाँ तक clear है? Is that clear? So now I will write down the 4 types of gametes here. Before I erase match with this, so that I write down the gametes here. What are the type of gametes now? Tell me. What is the first combination of alleles? Capital R and capital Y. What am I writing there? Gametes मतलब क्या? This particular chromosome forms one type of a gamete. So this will result in the formation of one type. This will result in the formation of other. This will result in the formation of other. This will result in the formation of other. That is what we are understanding. So abhi I have to write down the type of the gametes. What do I write? Capital R, capital Y, capital R, small r, capital Y, and small r and small y. Understood? Check.
What's the Y for the R?
Why not?
In the third gamete,
Ah.
capital letter comes before.
That also you can write. I prefer to write all the round alleles first. Is this understood? How many type of gametes now for this F1 plant? 4 types of
Why
gametes.
cannot be the R and R?
Which one? How can it be R and R? कौन सा? R and R कैसे होगा? R and R possible ही नहीं है. We are talking about every single chromosome. Now, see here, why the concept of independent assortment is loco-independent assortment क्यों कहा जाता है? After the process of all these crossing over and assortment done, every single chromosome goes in the formation of one type of a gamete. अब जब ये particular chromosome will enter here to form a gamete, is it dependent on the other 3? No, because every single chromosome has one allele for shape and one allele for color. Every single chromosome has one allele for shape, one allele for color. One allele for shape, one for color. It is like this. Why the concept of independent assortment? Why is it known as independent assortment? Every chromosome goes in the formation of a gamete independent of the others. This goes in the formation of a gamete independent of the other. This goes in the formation of a gamete independent of the others. Hence the concept of independent assortment. Then what do I understand? Dependent, when the different type of alleles were clubbed together, form something new, chromosome remain attached. तो एक क्रोमोसोम के साथ दूसरा क्रोमोसोम का एक साथ आ जाना, that is dependency. ऐसे कुछ नहीं हो रहा है यहाँ पे. Every particular chromosome, all the 4 chromosomes form 4 type of gametes. Independent assortment. So now again I write for this plant. What are the 4 types of gametes then? For this plant also. This is one. What is the other combination? This is one. What about the third combination? This is one. And this one. Are these the 4 type of gametes? Now, every plant has 4 types of gametes. Crisscross possible hai? The number of gametes are too many that we can carry out the process of crisscross. So we have to definitely go by the Punnett square method. So let us now try to understand the Punnett square method and get the F2 generation. All of you will write till here and then you will make the Punnett square. Write till here very neatly. This is just for understanding that I told you that you may write. No, for the cross in the exam you are not writing that. That is just for explanation that I told you. You are writing that within the bracket is just for explanation. Come on. Complete till here all of you and then make the Punnett square. The Punnett square that you make, it has to be a big one, not a microscopic one you make. The Punnett square that you make, since your notebooks are ruled books, 1, 2, 3, 4 and 5. 1, 2, 3, 4 and 5. Every single box should be at least minimum 2 to 3 lines. Depend on your handwriting. If your handwriting is a big one, 3 lines you have to keep. You have to write down the phenotype, the next line the genotype. It's like that. So don't make only one line box. This should be minimum 2 to 3 lines. This is minimum 2 to 3 lines. Remember, if your writing is small, then 2 lines enough. Then how do we go about? One plant we consider male, the other one we consider female, and keep writing down all the 4 gametes. What are the gametes? This is one, this is another, this is one, and this. Write down, make the Punnett square. Nice, neat, with pencil, make the Punnett square. Make this the tabular arrangement to get the F2 plants. Then next is I will write down the same gametes the other side also. And for ease of understanding, I mark them as 1, 2, 3, 4. This also not to show in the exam. Only for understanding.
We have written the same thing.
What the same thing?
Okay.
Got it? ये arrangement थोड़ा इधर उधर हो सकता है, उससे कोई problem नहीं. But see that you write all the 4 types. आया समझ में? All the 4 types. Now what are we going to do? What did we do in the previous Punnett square? Take one, fertilize with the other. 1 with 5, right here. 1 with 6, right here. 1 with 7, right here. 1 with 8, right here. Now that we are writing down the letters, what are we writing? Phenotype or genotype we are writing?
Genotype.
Genotype we are writing. So let us start. 1 with 5, what will I write? What is the genotype? 1 with 6.
R small r.
Got that? 1 with 7.
R small r capital Y.
1 with 8. Understood? Now first you complete with all the 3 more boxes, the 3 more lines there. This is done. Now take this one and go with it. 2 with 5, 2 with 6, 2 with 7, 2 with 8. Complete now the Punnett square. First you write down all the genotypes correctly. You make a mistake there. Why? What did I tell you just now? See the instruction that I repeated thrice. क्या बोला था मैंने? हाँ. Which one you are writing? हाँ. Make the box bit bigger. You write down the gametes. Why so clumsy? Neat लिखना है. Repeatedly I am telling you. Remember, ये जो crosses that you write when it comes for 5 marks, it is just like maths. Half mark का distribution रहता है. Half mark for writing down the phenotype, half mark for writing down the genotype, half mark for writing down the gametes, another half mark. So one step gone, you lose half marks there, 1 mark there. So हर एक step neat and nice and well explained लिखना है, so that you get full 5 on 5 in those answers. Next is, once you are done with this, you will have to complete with 2 and 5 and 2 and 6 and 2 and 7, 2 and 8. Yeah? Then we go with 3. Done?
Yes.
So 2 with 5, if I take capital R, capital Y, capital R, small y, complete करो till the 4th gamete, complete. Anybody has a difficulty in understanding? Is this understood? How the 4 types of gametes? Once you know how the 4 types of gametes, then making the Punnett square. Once you make the Punnett square, then the reading of the phenotype. That's it. Complete, all of you complete the Punnett square. What happened there? School exam?
Second July. When I
August.
say
August.
sorry. 10 July.
2nd of July.
Starting August.
2nd August starting.
But we don't
10
want
July.
science.
10 July.
तो science कब है?
Second 11 July. नहीं 12 July को. Third paper.
Ocean life process full है?
लाइफ प्रोसेसेस सारे 3 चैप्टर फुल है।
हाँ?
3 चैप्टर के फुल है। फिजिक्स केमिस्ट्री बायो।
एक चैप्टर ना? हाँ तो 40 मार्क का कि 50 मार्क का?
40 मार्क्स। बस बायो। अरे बस बायो तीन चैप्टर जाएगा।
3 chapter. Only 3 chapters.
हाँ।
August and then we have R and Y.
Group coordination नहीं आता? हाँ तो you are finishing.
We have R and Y.
इसके बाद करेंगे।
इसके बाद R environment है ना?
R environment में क्या पढ़ा? You know it all.
Life.
तो so simple है, वो तो कभी ना कभी पहले पढ़ा होगा ना थोड़ा सा। हाँ? वो पढ़ा नहीं था वो तुम्हारी गलती है ना। चलो आया समझ में? हाँ?
अरे वो लास्ट चैप्टर था ना बायो का।
Natural resource.
नेचुरल हाँ वही।
उसका ही तो आधा उसके कॉन्सेप्ट है। अच्छा हो गया ना? Third line is over. Don't copy.
हो गया।
I asked you what to do. Don't copy. You do. What are you supposed to? How are you filling up the boxes? 1 and 5, 1 and 6, 1 and 7, 1 and 8. करो. Don't copy. Why are you copying? That's why I have not done it all. Otherwise, most of people just copy down without understanding. Once you know the method I told you, based on the method you complete. Complete karo. Done? Fast, fast. बात कम कर फटाफट।
हो गया।
राजदीप नहीं आया समझ में? हाँ समझो ना, when you understand in the class, your effort becomes less at home. Done. Next, let us take this. I will write down the dominant first. So I will take this and this one, capital R, small y, and this, and you can write dominant first, not a problem. And the
ऐसे ही गया है सब लोग चलेगा.
it's okay once you have understood that मुझे इसके साथ इसका, इसके साथ इसका, इसके साथ, so that you can interchange and write also. There is no problem in that. There will be no mark deduction for that. And this one, check karo, is it correct? The letters that you write down, because if you are writing capital and small, mistake agar hoga, errors hoga, then the ratio will be all different. Complete hua?
Yes, ma'am.
Did you understand how to fill in the Punnett square? Rajdeep done? Did you understand how to fill in the Punnett square? Why are you looking at this book? Did you understand how to fill in the Punnett square? To karo na. If you have understood, then do on your own. It's so simple. Now tell me. Let us now start. Once you are done, make it fast. Done. Now we are going to. Now this is what we have written down, the genotype. Now we are going to write. Now we are going to understand the phenotype. So phenotype कैसे read करना है? To understand the phenotype, if I talk about a genotype like this, when we see one dominant allele is there, obviously the dominant allele will express. So is it necessary that always both the chromosome should have dominant allele to express the dominant allele? No. One dominant allele or dominant allele in only one chromosome is enough. And recessive trait कब express होगा? When both the traits in recessive trait, when will it be expressed? When both the chromosome has the same recessive trait, same recessive allele. Clear hai? So basically we are again talking about a concept of dominance. When will a dominant allele express? Say suppose, when will the seed be round? ऐसा है तो भी होगा round. ऐसा है तो भी round होगा. Round होगा. ऐसा है, then also round. Okay, let us try understanding. What about the phenotype of this one? The phenotype of this is round and yellow. What about this?
Round and yellow.
Round and yellow. What about the next?
Round and
Round
yellow.
Keep marking in your Punnett square now. Exactly this same method. Keep. नहीं, सारे कैसे round yellow हो जाएगा? Round and yellow. Go to the next line. Go to the next line. Tell me, what do I write here?
round and yellow.
What do I write here?
Round and
Round and
green.
green. I would write round and green. I write round and green. What do I write here?
Round and yellow.
Round and yellow. Who is talking? Round and yellow. What about this one? Round and green. एक और round and green बताओ कहाँ पे है? वहाँ पे कहाँ पे? Round and green.
उसके नीचे.
This one.
No,
Round and green कहाँ पे?
One.
Round and green. Where will you write?
नहीं. Ma'am.
Round and green.
Wrinkled.
Next. Round and green. One more will be there.
मैम आपने वो वाला गलत लिखा है मैम.
बताओ कौन सा?
2-8 वहाँ पे
8.
हाँ मैम वो वाला
Small
small
y,
y.
capital Y.
मैम 2-8 not 3-8. Small 2-8 मैम small y. Small y होगा मैम लास्ट में.
Correct. हाँ. So ये हो गया. Now the next is also round and green. Done. Now see the other one. What about the phenotype of this? Round and yellow. This is round and yellow. What about this one?
Round and yellow.
Round and yellow.
Wrinkled and yellow.
That's it. Do we have any more round and yellow here? Round and yellow. And now what about this one?
Wrinkled and green.
Wrinkled and
yellow.
yellow. Wrinkled and yellow. What about this one?
Wrinkled and yellow.
Wrinkled and yellow. Here again we have got wrinkled and yellow.
Wrinkled and green.
What will be the phenotype of this one?
Wrinkled and green.
Wrinkled and green. This is wrinkled and green. How many types of plants we got? How many types of plants we got? Four types. Count their respective number to find out the phenotypic ratio. So how many types of plants? We are now trying to write down the phenotypic ratio.
9:3:2.
The phenotypic ratio that we write is 9:2. Okay, let us go by the numbers first. How many round yellow plants?
9.
How many round yellow plants would be 9? What is the second type?
Round green.
Round and green. All of you. Round and green. How many?
3.
Round and green. How many? 3. Round and green 3. Wrinkled and yellow. How many?
3. And 1.
Wrinkled and yellow 3. And the fourth type is wrinkled and
Green.
green. Wrinkled and green. How many? 1. So what is the phenotypic ratio? The phenotypic ratio is 9:3:1. Genotypic ratio we are not studying, which is not there in your syllabus right now, but इसका भी genotypic ratio. हाँ, ये पूरा ये break करना पड़ेगा. Then that is the genotypic ratio that we understand. So now, is this clear?
Yes.
So now, how many types of plants we get? Here, when we were talking about the monohybrid crosses, there we were only talking about 4 plants, and out of the 4 plants, the ratios. Here, how many plants we are talking about? 16, and the ratios on 16 plants. How many types? 4 types. Phenotypically 4 types. So that's the reason we are writing down this ratio 9:3:1. Now, this is the entire explanation. Give an example of give an example of a dihybrid cross. Give an example of a dihybrid cross. With any pair of alleles, you can give an example. Instead of round and yellow, what else can I take? Tall and axial. I can take tall and purple, yes. So if I take tall and purple, then what should I take here? Dwarf and white. Okay. So that way, sometimes you straight away get this. As I told you, there will be 2-3 types of questions on this. Sometimes straight away, state the law of dominance, state the law of independent assortment, explain with the help of an example. So if that is the condition, then straight away this particular example you can explain. But sometimes questions will be given and you will have to answer accordingly. Once you are done with this, I will give you one question. But before that, let us write down the statement of the law. Independent assortment. Law for 1 mark, what to write. What is the statement of the law? What will we write down for 1 mark? Write down independent assortment. Within inverted commas, again you are supposed to write, as we have done it earlier also, for the law of dominance and segregation. My God, the notebook का हाल तभी ऐसे कैसे होगा? 4 बार बोलने के बाद भी. Now, write down the statement of the law. When parent plants When parent plants with two or more contrasting traits. When parent plants with two or more contrasting traits slash alleles भी लिख सकते हो, that is optional, anything we can write. When what I say, when parent plants with two or more contrasting traits or alleles are crossed, traits or alleles are crossed, inheritance of देखो उसका बॉक्स कितना अच्छा बनाया उसने, inheritance of, हाँ छोटा नहीं है, छोटा नहीं है, हाँ it is neat, but थोड़ा एक लाइन और स्पेस रखते तो और ज्यादा अच्छा होगा. Inheritance of one pair of allele, one pair, ये one pair है कि नहीं? One pair है, one allele for shape, one allele for color. Inheritance of one pair of allele is independent. Inheritance of one pair of allele is independent. Pair of allele is independent of the other pairs. Independent of the other pairs during gamete formation. Inheritance of one pair of allele is independent of the other pairs during gamete formation. आया समझ में? आया समझ में? That is it. For the law, that much only. Now you take one question and I want to see how many of you have understood. Fatapat likho, let me discuss the answer right now. In a dihybrid cross, question in a dihybrid, did you understand something? In a dihybrid cross, homozygous, tall plants with purple flowers. Tall plants with purple flowers are crossed. Tall plants with purple flowers are crossed with dwarf plants and white flowers. Full stop. Dwarf plants and white flowers. Full stop. On selfing, मतलब यही self pollination. F1 plants को जो selfing करते हैं. On selfing, you will get this word in your paper. SELFING. On selfing, F2 plants were obtained. On selfing, F2 plants were obtained. ये हो गया question. Now you write down the सब questions जिसके आंसर्स लिखने हैं. Number 1, अभी सब number 1 लिखो. Number 1, what is the phenotype of F1 plant? What is the phenotype of F1 plant? एक लाइन गैप छोड़ो. Number 2, which allele, पहले लिखो question, which allele or trait in the F1, which allele or trait in the F1 is dominant? One line for the answer. Number 3, how many types of gametes? How many types of gametes will the F1 plant produce? How many types of gametes will the F1 plant produce? 2 lines for the answer. Number 4, how many plants in the F2 generation how many plants in the F2 generation have the parental phenotypes? How many plants in the F2 generation have the parental phenotype? 2 lines for the answer. Last, number 5. How many plants in F2 generation are recombinants? Recombinants. How many plants in the F2 generation are recombinants? Recombinants, question mark. Next, continue. Give one reason for such combination of alleles. Give one reason for such combination of alleles. Give one reason for such combination of alleles. Now, ये हो गया दूसरे तरह के क्वेश्चन. और भी आता है with numbers that we will do later. No class work. फटाफट करो. Go through the question. अब देखो ये full 5 mark का question है. Are you doing the Punnett square? Punnett square करने के लिए कोई मार्क है? Show with the help of a Punnett square लिखा है question में? No. तो Punnett square करने के लिए कोई मार्क actually नहीं है. But if you are not very thorough, Punnett square करके तुमको आंसर लिखना पड़ेगा. Don't just guesswork and write. It may be wrong. Once you are thorough with this concept, then you can easily write down the answers. Now let us try. Those of you who have understood, you will be able to write down. Chalo. What is the answer for the first question?
Hybrid tall purple.
What did I ask you? Read the question. Wait, one by one. Let me check how many of you have understood this. Number.
Phenotype of F1 plant.
हाँ. क्या होगा? The crosses between tall and purple, dwarf and white. So F1 का phenotype क्या होगा? Will take 2 minutes. Don't allow your brains to get lost. Yes. F1 का phenotype क्या होगा? Next, F1 का phenotype क्या होगा? The crosses between tall purple, dwarf white. तो F1 का apply करो ना ये concept. F1 का phenotype क्या होगा? अभिषेक, F1 का phenotype क्या होगा?
Hybrid tall white plant.
Hybrid
Tall white.
white कैसे हो जाएगा? Hybrid white, hybrid tall and purple. F1 में हमेशा dominant traits ही express होता है. Hybrid tall purple. Next question.
Tall mama.
Next question.
Which allele in the F1 is dominant?
Which allele in the F1 plants are dominant? Which are the dominant alleles?
Purple.
Purple and tall. Tall and purple alleles are dominant. Likho. The next class again I will give you this. Go home and go through. Tall and purple dominant है. Number 3. A. वाग्मय. Number 3 बोलो. हाँ. You. हाँ. बोलो. Number 3.
How many types of gametes will the F1 plant produce? 4.
4 types of gametes. Yes. आया समझ में? 4 types of gametes in the F1. Next बोलो. Question number 4.
How many plants in F2 generation recombine and give one?
नहीं.
Fourth question. How many plants?
How many plants in the F2 generation show parental phenotypes I gave you? How many plants out of the 16? How many plants in the F2 generation show parental phenotypes? कितने हैं? Parental phenotype किसको consider करेंगे? Parental phenotype is
10 आते हैं. दोनों हो जाएंगे ना.
हाँ. आया समझ में?
10 हो जाएगा.
How many of you understood this? कैसे 10? ये हो गया parental and ये हो गया parental. है ना? Parental. So how many plants? Out of 16, 10 हो गया. Next, आया समझ में? तो बाप में से थोड़ा दूरी बना के रख तो दोनों को समझेगा। आया समझ में? बाप में तुमने क्वेश्चन पूरा लिखा कि नहीं लिखा अभी तक? आंसर कब लिखोगे? नेक्स्ट नंबर 5 का आंसर क्या है? रिकॉम्बिनेंस मतलब रिमेंबर आई एक्सप्लेन यू हियर न्यू कॉम्बिनेशन ऑफ एलील्स। कौन से तरह का प्लांट है जो ये वाला फेनोटाइप नहीं है? which are the plants? ये एंड ये सुनो, पैरेंटल था राउंड एंड येलो, अब यहाँ पे आ गया राउंड एंड ग्रीन। पैरेंटल था रिंकल्ड एंड ग्रीन, यहाँ पे आ गया रिंकल्ड एंड येलो। तो ये न्यू कॉम्बिनेशंस है जिसको रिकॉम्बिनेंट्स बोलेंगे। नंबर इज 6। रीजन, एक रीजन लिखना है, क्या लिखेंगे? व्हाई recombinants have happened due to the process of crossing over। देखो लास्ट लाइन, recombinants occur due to the process of crossing over, due to the process of crossing over and exchange of chromatids। Recombinants occur due to the process of crossing over and exchange of chromatids। आया समझ में? एक्सचेंज ऑफ क्रोमोसोम्स में लिख सकते हो, एक्सचेंज ऑफ क्रोमोसोम्स या क्रोमैटिड्स। तो क्रोमोसोम सेगमेंट का अरेंजमेंट की वजह से नाउ द रिकॉम्बिनेंस। आया समझ में? गो थ्रू ऑल ऑफ यू दिस पर्टिकुलर कॉन्सेप्ट, फाइंड आउट सम टाइम दो दिस चैप्टर इज नाउ मे नॉट बी देयर इन योर कमिंग एग्जाम, but still is it there? Go through this at home. Again I've got a class with you on Wednesday, so I'm going to finish with this chapter and start with the next. So and on top of that समझ में नहीं आ रहा, बता ये वाला चीज आरो पकड़ के आया? समझ को आया? Next class I have got full 2 hours class. I'm going to take a oral on life process and little bit of control coordination also। control coordination अभी एग्जाम में नहीं है, पढ़ाया भी।
मैम दोनों नहीं
सुन अनिल, काम के बाद अरे काम था, सही में काम था। सुन
सेम ही है भाई, वही तो चिंदी नहीं है।
टेक्स्ट बुक मस्त है, इन लोग का सब कुछ एक है, इन लोग का है।